Alrighty, so this is going to be our detailed aerobic respiration video. So this white sheet of paper is going to be our animal cell. So let me draw the cytoplasmic membrane here. This is an oddly square-shaped animal cell, but there you have it. The same aerobic respiration process also happens in plants, in bacteria, in protists, etc., sometimes with a couple tweaks. But in general, it's the same process. But since you guys are learning about animal cells in GenBio, we are going to do the animal cell version, which is the standard version, so it will match what you've learned before. So this is going to be our cytoplasm of the animal cell, and then I'm going to draw one enormous mitochondrion because a lot of stuff is going to happen in the mitochondrion, and so I want to be sure that we can see it. Okay. So this is our mitochondrion, this is the outer membrane, this is the inner membrane. This space in here is called the intermembrane space, the space between the two membranes. And then the inside of the inner, so this is the inner membrane, the inside of the inner membrane is called the matrix. So hopefully you guys are now all oriented to our animal cell with its one gigantic mitochondrion. There bizarrely are some protists that have one gigantic mitochondrion, but animal cells do not. Okay, so back to the thing. Let me zoom in a little bit. So what is the first step? You guys all know this. So you eat, and then you use your salivary amylase in your saliva to break down the starch into sugar, and then it turns into glucose. And then our first step is going to be glycolysis, wherein we will change our 1,6-carbon glucose into 2 3-carbon pyruvates. This is a multi-step process. I think that it is 10 steps. For a lecture, Dr. Knight will want you guys to learn each of the steps. For lab, you'll just need to know what I am putting in the video, which is not going to be each of the 10 steps, but nevertheless, there are 10 steps. So we need to add 2 ATPs to get glycolysis started, and at the end, we are going to make, I'm going to write a minus because I think that'll help. We're going to make 4 ATPs, so we end up with a net production of 2 ATP in glycolysis. We're going to circle that with the yellow. Hopefully you guys can see that. Then, because we have broken the bond, so you guys remember we had the long 6-carbon glucose, and then we broke that middle bond to turn it into 2 3-carbon pyruvate. Because we have broken that bond, we have released some electrons, and those released electrons are always going to be picked up by an electron carrier. And so we have made 2 NADHs. We do not meet the FADH2 until the Krebs cycle. So to start with, we're just going to be NADH. So where is this happening in our animal cell? It is happening in the cytoplasm. So glycolysis happens in the cytoplasm. 1 6-carbon glucose to 2 3-carbon pyruvates produces 2 NADH and a net of 2 ATP. So from that, we're going to take our 2 3-carbon pyruvates and use them in the next step. And so here is our 3-carbon pyruvate. Anytime that you have more than 1 carbon together, there is still some energy that can be gained. So 1 carbon by itself is just CO2. We cannot get any more energy from that. We just breathe it out as a waste product. Anytime that there are multiple carbons connected, then energy can still be extracted from that by the body. And so in a little bit, we're going to, in a separate video, not in a little bit, in a separate video, we're going to talk about fermentation and the reason that if you drink a lot of alcoholic beverages, it can cause you to gain weight is in the alcoholic beverages, there is still some connected, ethanol is a 2-carbon compound. There's still some connected carbons. And so as long as you have connected carbons, there's energy remaining there. And so we will continue to work with these to break them down into the individual CO2s to get out all of the energy that we can. So we're going to take our 2 3-carbon pyruvates and we are going to actually move them into the mitochondrion into the intermembrane space, the space between the 2 mitochondria. And so there we're going to do pyruvate oxidation. And again, oxidation is losing electrons. And when you lose electrons, you break bonds. Bonds are held together by electrons. So when you break a bond, you lose electrons. When you lose electrons, you break a bond. Pretty much saying the same thing. So this is going to be our pyruvate. We're going to break this bond here. And so, because we actually have 2 pyruvates, right? And so we're going to start with 2 3-carbon pyruvate. We're going to release, for each pyruvate, we're going to release 1 CO2, but since we have 2 of them to start, we're going to release 2 1-carbon CO2s. And then our remaining 2-carbon piece is going to be an acetyl group, and it is going to be joined by coenzyme A to form 2 2-carbon acetyl-CoA. Okay? This is what we're going to use in the next step because it still has 2 carbons connected, so it's still going to have some energy. Because we had to break a bond to release this CO2, then we are also going to make 2 NADHs from our 2 pyruvates. So then our 2 2-carbon acetyl-CoAs are going to come into the matrix of the mitochondrion, and that is where the Krebs cycle will happen. So the Krebs cycle is also called the citric acid cycle, the tricarboxylic acid cycle, the TCA cycle. You might have heard one of those other names. I'm pretty sure that Dr. Knight calls it the Krebs cycle in class, and so that's what we're going to call it in this video. So the Krebs cycle takes the 2 2-carbon acetyl-CoAs, and a lot of stuff is going to happen to these guys. So these 2 carbons are actually going to match up with 4 other carbons that are already in the cycle, and there's going to be a whole bunch of rearranging and a bunch of different reactions, which you will not need to know any of those. What you are going to need to know is that these 2 carbons here are going to come apart. So since we have 2 2-carbon acetyl-CoAs, we're going to, by the time we break both of those apart, we're going to release 4 carbon dioxides, which are going to be breathed out. And so at this point, we have completely dismantled our 6-carbon glucose into 2 CO2s here and 4 CO2s here. So we've broken down our 6-carbon glucose into 6 CO2s, all of which we are going to breathe out. Because there's so many different steps in this cycle, a lot of other stuff is also made. So we are going to make 3 NADHs for each turn of the cycle. But again, the cycle will happen twice, and so that's going to be a total of 6 NADHs. We're going to make 1 FADH2 each time the cycle goes around, but the cycle will go around twice for each glucose. So we're going to make 2 FADH2s, and then we are also going to make 1 ATP for each turn of the cycle, but the cycle will happen twice, so we end up with 2 ATPs. The cycle actually, the Krebs cycle actually makes GTP, but GTP is energetically equivalent to ATP, and so, generally speaking, and so the 2 GTPs that are produced are usually lumped into the ATP total instead of making a separate GTP only category. Okay, so the Krebs cycle is going to happen in the matrix of the mitochondria. It finishes taking apart the glucose to release 4 CO2s and produces a total of 6 NADHs, 2 FADH2s, and 2 ATPs. So it's really important for you guys to realize that at this point, we have completely broken the glucose. So the electron transport chain is not going to have anything to do with the carbons. They're all CO2s, and we have breathed all of them out. What we have made is a bunch of electron carriers. We've made some NADHs and we've made some FADH2. And so the electron transport chain is going to take the electrons and the energy from the NADH and the FADH2 and use that to power the formation of ATP. So the electron transport chain, what color do I have left? We will make it orange. The electron transport chain happens in the inner membrane. It is physically located in the membrane of the mitochondrion. And I found a really excellent drawing online. It is a still from a Khan Academy video. And so I've printed that out, and I'm going to walk through that with you guys, but for the electron transport chain. Okay, so we'll just set this up here. And so here is our electron transport chain. Okay, so again, this whole thing is going to be our cell. This is our giant mitochondrion, and this is part of the inner membrane here. And so outer membrane of the mitochondrion, inner membrane of the mitochondrion, intermembrane space between the two, and then the matrix of the mitochondrion is right here. So hopefully you guys feel pretty oriented. Okay, one of the really important things that Dr. Knight is going to want you to know is the NADH and FADH2 conversion to ATP. Okay, so back to this picture for just a second. These two NADHs, because they are made in the cytoplasm, some energy is lost in transferring them into inside the mitochondrion in order to be in the electron transport chain. So these are going to make 2 ATP each. Where's my purple? So from these two, we're going to eventually get 4 ATP. Okay? So our NADH cytoplasmic, produced in the cytoplasm, is going to give enough energy to form 2 ATPs each. Our NADH that is formed anywhere in the mitochondria, so in the intermembrane space or in the matrix, is going to make 3. It's not doesn't directly make 3 ATP, but the NADH produced in the mitochondria give enough energy to make 3 ATPs. And then the FADH2, which is only produced in the mitochondria, gives enough energy to produce 2 ATPs. So Dr. Knight is going to want you guys to know this conversion, and this is what it is. Cytoplasmic NADH, 2 ATPs each. Mitochondrial NADH, 3 ATPs each. And mitochondrial FADH2 is 2 ATPs. So you might be wondering why the mitochondrial NADH and the mitochondrial FADH2 are different, and this is why. So here is our electron transport chain. It's physically located in the inner membrane of the mitochondrion. And it has, you guys have always seen this, right? Drawn as like stair-step situation, maybe. And so there are 4 different complexes, sorry, 1, 2, 3, 4, in the, this is a separate thing, in the electron transport chain. And so you can think of them, if you learned the stair-step method, as the first stair, the second stair, the third stair, and the fourth stair. So the sooner in the process that an electron carrier enters, the more energy will be available from those electrons. So NADH comes in at complex 1, and so it releases enough energy to form 3 ATPs. Again, this is mitochondrial NADH. Cytoplasmic NADH only releases enough energy for 2 ATPs because it takes some effort to get into the mitochondrion. But then the FADH2 is going to enter and donate its electrons to the second complex of the mitochondrion, And so since it joins the process later, like kind of on the second step, it does not release as much energy. So it only makes 2. So mitochondrial FADH2 gives enough energy for 2 ATPs. Okay, so our NADHs, which are going to be made in pyruvate oxidation, glycolysis, and the Krebs cycle, and our FADH2s, which are only made in the Krebs cycle, will not all, there's only 2 of them, will both donate electrons to the electron transport chain. As electrons go down the electron transport chain, they will push protons into the intermembrane space, the space between the 2 membranes. And so the important thing to remember about protons is that they are charged. And so you guys have learned about the membrane and the fluid mosaic model, and you know that charged things do not easily go through the membrane because of the nonpolar tails. And so the protons are stuck here between the 2 membranes. And so what they are wanting, they want to be less stuck. Okay, so if you think of this, you know, pre the Amazon days as like Black Friday shoppers, or if you're like trying to get into a really good sporting event, or anywhere where there is an enormous line and one tiny gate. Like everyone is kind of smushed together in the line, but they really just want to go through the gate so they can spread out and do the thing. So that is this. And so as the electron carriers donate their electrons, the electrons go down the electron transport chain, protons are pushed, transported into the intermembrane space. Eventually, these protons accumulate and you get a high concentration of protons in the intermembrane space. And the protons would really like to be more spread out. They feel kind of smushed. They don't get along that well because they're all charged, and now they're like grumpy because they're together, anthropomorphizing the protons. So what they really want is to be able to find a little door through the membrane so that they can go through. And so that's what happens here. And so this is the enzyme, the protein, the enzyme called ATP synthase. And it is the awesomest enzyme ever because it spins like a little propeller or like a little water wheel or a turnstile or anything that you can think of that spins. And so the protons will go through. It takes 3 protons to make each ATP. And so, right, so you've got your adenine, and then it's got 2 phosphates on it. Phosphates are negatively charged. So you guys know from like your little kid magnet experiment, right? Like the 2 ends of the magnet that are the same charge don't want to go together, right? You like try to push them and they push apart. This is the same thing. So just like how the protons did not want to all be smushed because they're all positive, it takes a huge amount of effort to add this 3rd negatively charged phosphate to the 2 that are already there. And so it almost like screws it on. And so the protons come through the ATP synthase, and then this extra phosphate is added, and it's kind of like until it sticks on. And that is what makes ATP so high energy. So the second that this bond is broken, this 3rd phosphate is like, whee! And it springs off because it's negatively charged, so it doesn't really want to be there anyway. And releases a whole bunch of energy. So that's probably got a little bit sidetracked there. So the electron transport chain is going to happen in the intermembrane space. No, sorry, happens in the inner membrane. The electrons that are donated by the electron carriers will enter the electron transport chain because the NADH electrons and the FADH2 electrons enter at different steps. They release different amounts of energy and can make different amounts of ATP. Each of the electrons will use the energy that it is releasing to pump protons, to power the pumping of protons by these different proteins, but to pump protons into the intermembrane space. And then that accumulated concentration of protons will go through the ATP synthase to power the ATP. Woohoo! We have made ATP. All right. Why is this called aerobic respiration? Well, this is like a, kind of a chain. You pass the electron to the next complex. If there's nowhere for the electrons to go in the long run, then the chain just gets backed up, and then we get no protons pumped across, and then we get no ATP made. And so we need a place for the protons to go. We need a place for the electrons to go so that the electrons can go through the electron transport chain, and then more electrons can be taken from the NADH and the FADH2. If these electrons cannot be donated, then, let me zoom out for just a second. None of this other stuff can happen, right? Because you have to have 2 NAD pluses in order to make your 2 NADHs, etc. And so if this electron does not get donated and this NAD plus does not get made, then the whole operation stops and you get 0 ATP and then you die. And so we need a place for the electrons to go so that the whole process can continue. And in aerobic respiration, the electrons go to oxygen and then water is formed. Technically, oxygen is only used in this very last step. So we have not used oxygen in any of the previous steps. The dismantling of carbon does not specifically require oxygen. Oxygen is only used in the electron transport chain of aerobic respiration. So this water that is produced here, fascinatingly, I think, is called metabolic water, and is the reason that you have to go to the bathroom even if you don't drink a lot, because so you breathe in oxygen. As you are breathing in oxygen and just sitting there, your body is just turning the oxygen into water in the process of doing the electron transport chain so that ATP can be produced. Okay, I'm sorry, I got super excited and that was kind of long, but that's okay.